Hi dear students. So today let's talk about culture media. So let's see the definition of culture media. A growth medium or culture medium is a liquid or gel designed to support the growth of microorganisms or cells or small plants like the moss, Phycometrella patterns. Or in other words, we can say that any liquid or solid preparation made specifically for the growth, storage or transport of microorganisms or other types of cells. Let's check into the history of culture media. Robert Koch could be considered the father of culture media. His first success in bacteriology was in the isolation of Bacillus anthracis, which at the time was causing the disease anthrax in cattle. This was the first time that any pathogenic organism had been isolated and studied outside of the host's body. It was after this discovery that Koch wrote his famous postulates regarding the causative agent of infectious disease, which were published in 1882. According to that, the organism must be consistently associated with disease. It must be isolated and grown in pure culture. And the pure culture must be shown to induce the disease when injected into experimental animals. Although Louis Pasteur and Koch are both considered to be responsible for the establishment of the science of microbiology, Koch and his co-workers systematically identified all the agents of the major bacterial infectious diseases of the 19th century. Even though many causative agents of disease have been successfully discovered, uh, the ideal medium was still not available. Since a practical solid media had not yet been discovered, he used broth made of sterile beef serum or aqueous humor taken from the eye of an ox, and he also observed the growing and sporulation of organisms directly under the microscope on a warmed stage. Other scientists had experimented with various substrates such as bread, potato, coagulated egg, galvanine, starch, paste, and meat for growing bacteria. And Koch also observed that few pathogenic microorganisms would grow on potato and other media such as egg. Credit must also be given to the Italian scientist Bartolomeo Bizio, whose work preceded Koch's by 50 years. He was possibly the first to attempt to grow an organism on a solid medium. In 1832, there were reports of the miraculous appearance of blood spots on bread and communion wafers. Bizio was able to subculture a chromogenic bacteria onto other bread surfaces in pure culture, and he named it as Serratia marcescens. And partial success was reached when Koch discovered the use of gelatin that could be mixed with broths and bouillons. In 1881, he presented his technique at the International Medical Congress in London, which was attended by Louis Pasteur and Joseph Lister. And there were two discouraging problems associated with the use of this new nutrient gelatin medium. The first one is that it changed from a solid to liquid at about 25 degrees Celsius, preventing it from being incubated at 37 degrees Celsius, which is optimal for most of the bacteria. And the second one is that the gelatin is liquefied by the enzyme gelatinase, and this is also produced by most proteolytic bacteria, and that is, this enzyme is also produced by most of the proteolytic bacteria. At last, in late 1881, all the problems were soon overcome when agar was tried out as a solidifying agent. And credit for this discovery belongs to Fanny Hesse, who was born in New Jersey. And Fanny married the German physician Walter Hesse, who was experimenting with airborne bacteria in Saxony. Fanny, who served as Walter's laboratory technician and illustrator, pondered the problems associated with gelatin-based media and suggested using agar instead. Fanny and her mother had used this agar to prepare fruit jellies. Even now we are using agar for preparing fruit jellies and puddings. And there it had been used for generation as a gellifying agent, which was useful in hot climates. So what is agar? Agar is a polysaccharide that is derived from red seaweed. Its ability to melt at 85 to 95 degrees Celsius and to solidify at 34 to 42 degrees Celsius make it ideal for culture media. Another benefit is that it is clear for ease of observing colonies and was found not to be toxic to the bacteria. So this was found to be the best medium for culturing organisms. When Dr. Hesse visited Koch's lab for several weeks, he introduced him to the agar-based media, and Koch immediately recognized the value of this new agent and began to use it routinely in his work. Unfortunately, Fanny Hesse was not recognized for her contribution to microbiology until 1939, 5 years after her death. The simple creation of the sterile, transparent agar-based medium revolutionized and accelerated the research of that time and remained the basis of all practical bacteriology and mycology today. Now, bacterial colonies could be easily incubated, isolated, enumerated, and consequently characterized and identified using this medium. And this was the golden age of medical bacteriology, as Pasteur and Koch identified the bacterial origins of one disease after the another. So after agar was discovered, it was a common practice at that time to pour the media into a flat glass plate and then cover it with a bell jar while it is cooled. In 1887, another associate of Koch by the name of Richard Petrie decided to modify this method by putting the medium into a glass dishes with sides that were about 50 mm tall and a diameter of about 100 mm. The original design of this, that is Petri dish, has not changed up to this day except for being made of disposable plastics. So the Petri dish or the Petri plate that we are using even today to pour the agar medium was discovered by Richard Petrie. Let's see what this peptone is. Koch and his associate continued to experiment with bacteria using meat infusions or extract as their nutrient base. Although this is an adequate source of most growth factors for bacteria, it lacks sufficient concentration of amino nitrogen for optimal growth. It was Frederick Loeffler, who was investigating Corynebacterium diphtheria, who first had the idea of adding peptones to the nutrient broths. Peptones are short chains of amino acids that are prepared by digesting meat with enzymes. Loeffler also added sodium chloride to the medium, and this created the proper osmotic level for optimal growth. Negley found out that the peptones were the most effective of the nitrogenous compounds tested in promoting the bacterial growth. Also, the amino acids being too small and protein too large to be utilized by some bacteria, the mid-range size of the peptones appear to be the ideal source of nitrogen. And these peptones produced by protein hydrolysis are still the most significant ingredient in modern bacteriological culture media. Later, Emanuel Merck of Germany began to manufacture this peptone in 1892, and which gave the European microbiologists the option to purchase their media ingredients rather than fabricating their own. And now it's available everywhere. America's first media company was founded in 1895 and it is known as Digestive Ferments Company, and today it's known by the name DIFCO. Bactopeptone produced in 1912 was the first product aimed at developing the microbiological market, and he also pioneered the use of pepsin, pancreatin and trypsin enzymes for the digestion of plant and animal protein. Both Merck and DIFCO began using a new technique to produce the media in the dry powdered form, and this was done by spraying the media under pressure, then drying until a fine powder is formed. So there we concluded with the history of culture media. So now let's see the important factors required for the cultivation of microorganisms. First one is the proper nutrients must be available, oxygen or other gases must be available. Moisture is necessary. The medium must have an appropriate pH and proper temperature must be maintained. The medium must be free of interfering bioburdens and contamination must be prevented. And a satisfactory microbial culture medium must contain the available sources of carbon, nitrogen, inorganic phosphate and sulfur, trace amounts of metals, water and vitamins. They were originally supplied in the form of meat infusions. B4 yeast extracts frequently replace meat infusions in culture media. Meat infusion refers to the extract of B4 meat. Then the addition of peptones, which are digest of proteins, provides readily available sources of nitrogen and carbon. The pH of the culture medium is also important depending upon the microorganism that is growing on it. Temperature is another important parameter. There are different types of microorganisms. Some microorganisms are heat-loving and the other ones are cold-loving. The mesophilic bacteria and fungi have an optimal growth of temperature between 25 to 40 degrees Celsius. The thermophilic ones or the heat-loving ones are the grow at temperatures greater than 45 degrees Celsius. And the psychrophilic ones are the organisms that require very low temperature, that is below 20 degrees Celsius, and they are called cold-loving. And most of the human pathogenic organisms are generally mesophiles because they can survive in temperatures that is related to the humans, that is about around 37 degrees Celsius or in the range of 25 to 40 degrees Celsius. Let's see some of the common media constituents. The source of amino nitrogens are peptones, protein hydrolysate infusions and extracts. And source of growth factors are blood, serum, yeast extract or vitamins, or NAD, that is nicotinamide adenine dinucleotide. The source of energy are sugar, alcohols and carbohydrates. And buffer sources are the phosphates, acetates, citrates. And we can derive salt and metals from phosphate, sulfate, magnesium, calcium, iron. And there are also certain selective agents whose sources are chemicals or antimicrobials or dyes. And indicator dyes sources are phenol red or neutral red. And the gelling agents that are commonly used are agar, gelatin, alginate, silica gel. Purified water is recommended for use in the preparation of culture media. It contains no added substance in it. Peptone protein hydrolysates, infusions and extras are the major source of nitrogen and vitamins in culture media. Peptones are water-soluble ingredients derived from proteins by hydrolysis or digestion of the source material. For example, by digesting meat or milk. Carbohydrates are also employed in the culture media as an energy source, and this can also be used for differentiating the genus and identifying the species of a particular organisms. Buffers are used to maintain the pH of culture media. Selective agents include bile salts, dyes, and antimicrobial agents. Bile salts and deoxycholate are selective for the isolation of gram-negative microorganisms, inhibiting gram-positive cocci. So this can be used for the selective identification of certain bacteria. Then we can use dyes and indicators. These are essential in the preparation of differential and selective culture media. These are especially used for differentiating different kinds of bacteria in the time of identification. In this formulation, these dyes will be acting as a bacteriostatic agent, that is, it will prevent the growth of a particular group of bacteria in order to promote the growth of the other group of bacteria which is of our interest. And these indicators may change the color when it is acidic, when the product that is produced by the bacteria is acidic or alkaline. Depending upon that, it will change the color. If a particular organism is capable of producing acid in the substrate, the indicator will change the color accordingly. Then we use antimicrobial agents in the media to inhibit the growth of bacteria, yeast and fungi. That is for the selective growth of a particular group of bacteria, and to avoid contaminations we can use antimicrobial agents. Then there is solidifying agents including agar, gelatin and albumin, and can be added to the liquid medium in order to change the consistency to a solid or a semi-solid state. Chromogens and fluorogens are also used to allow for differentiation and identification of the organism. When these substrates are degraded by a specific enzyme that is produced by the microorganism, they release differently colored or fluorescent compounds. For example, a MUG, that is MUG, is an example, that is 4-methyl lumbrylphenol beta-D glucuronide.